Okay, so thanks for joining everyone. So this is our first Theory of Living Matter seminar of Lent term. And today's speaker here with us, we've got Stanislav Shvartsman from Princeton University and the Platine Institute. And his work uses experiments, theory, and computational approaches to develop predictive models of dynamical processes in cells and tissues. And the current themes of his research include multiple aspects, I guess the physics, developmental biology interface, including the dynamics of signaling networks and the quantitative biology of developmental abnormalities. And so today he'll tell us about how signaling systems respond to perturbations. Thanks for joining us.
Okay, Joel, thank you for the introduction. Thank you for inviting me. Happy to be here. So the Zoom seminar brings back painful memories of COVID, but I'll try to ignore. Okay, so I will tell you about our work that has to do with responses of signaling systems to perturbations. The work is motivated by our attempts to develop quantitative thinking about developmental abnormalities. I will tell you today about why we are interested in this work and try to convince you that ultimately understanding this class of diseases depends on our ability to predict how signaling systems or biological systems in general respond to very specific perturbations. So first of all, as you probably know and hear from your developmental biology colleagues, embryonic development and biological organism development in general is an epitome of robustness and reproducibility. We know that dogs come from dogs, frogs come from frogs, but sometimes development does not go as planned and organisms are born with congenital abnormalities, and abnormalities can also develop and get more severe during postnatal development. So statistics is not very solid, but according to the Center for Disease Control in the US, 1 in every 6 US children has one or the other developmental abnormality. There is very limited funding for studies of these diseases because it's hard to come up with large cohorts of patients that can be statistically examined. So there is not much funding. There is even less quantitative understanding. Nevertheless, these diseases provide a window in the robustness and evolvability of development, because it is really from the variations of strongly canalized developmental trajectories that diversity of organism morphologies arises. So we need to understand this class of abnormalities and this class of diseases, both to understand how to manage them, how to predict them, how to prevent them, also to understand what gives the tell you about two sets of results, both of which have to do with perturbation of the same signaling system at the same signaling node. So there will be two types of perturbations. One will be permanent, and these will be perturbations that come from the side of the genome, which is something that is happening in individuals with genetically induced developmental abnormalities. And so in this case, the perturbation will be present in the organism from the moment of the formation of the embryo and throughout its lifetime. And the second class of perturbations will be acute, where the organism will be unperturbed up until a certain point of development, and it will be a pulse of light that perturbs a specific node on the signaling network. In both cases, we will look at the functional consequences of these perturbations for the unfolding of developmental programs. So humans knew about developmental abnormalities and described them already in antiquity. You can find them in Aristotle's writings, and even before Aristotle, there were books that catalog different types of deformities and tried to interpret them in terms of what this can mean and whether gods like you or do not like you. But there was absolutely no understanding of any kind. So it was not known whether this is the message from gods or whether this is something that comes from pure chance. So the first insight was provided by a French polymath, Maupertuis, and it is remarkable that this was done 100 years before Mendel. So Maupertuis, at some point in his career, was president of the Academy of Sciences in Berlin, so it was just newly established. When he arrived in Berlin, he found out about a surgeon who had the 6 fingers on both of his hands. So what is more interesting, so there are 2 things that are interesting. So first of all, that this person was a surgeon. Second, this surgeon knew that polydactyly, or the presence of extra digit on both hands, was present in 3 generations. So Maupertuis took the information about the incidence of polydactyly in a general population and did correct probabilistic calculations, something that you the first couple of undergraduate class. He concluded that emergence of polydactyly consecutive generation would be impossible chance in every. So concluded that this is it. Remarkable experiment. Cats the same trait, animals genetic. So this was that it congenital formation of one formed digit can be genetically transmitted. Of course, it was absolutely unclear what is it that was transmitted. Today, we understand that what was transmitted in this case is a specific mutation in the regulatory region of a gene that patterns developing limbs. What is absolutely remarkable is that something that is perturbed locally and gives a field that gives rise to a digit eventually gives rise to correct formation of muscles and nerves and bones, and even connects this fully functioning finger to the brain and gives it a cortical representation. So today, if you do a search for the words polydactyly and surgery, you will find doctors that will offer services for the removal of extra digits. So it's absolutely amazing that this person was a surgeon. And it turns out that there is some number of studies that describes that there are some motor activities of humans where it can be beneficial to have an extra finger. And apparently the Ruche, the surgeon that Maupertuis described, had this ability. So today we know that developmental defects can be monogenic and polygenic. So polydactyly is a monogenic developmental defect. They can be induced by mutations in coding and non-coding sequences. Polydactyly is induced by a mutation in a regulatory or non-coding sequence. We know that these mutations alter molecular and cellular processes in embryos. So we know a lot of mutations that are associated with developmental abnormalities. At the same time, there are lots of things that we do not know. So first of all, we do not know anything quantitatively about the magnitudes of the perturbations of molecular and cellular processes that give rise to perturbations. So we know as a consequence, we know very little about the sizes of the basins of attraction of normal developmental trajectories. We understand that these mutations perturb rate constants, induce patterning changes. In most cases, these changes have not been quantified. Furthermore, and this is a very important question these days, we know very little about what controls the penetrance and expressivity in these developmental abnormalities. Why is this a pressing question? It is a pressing question because as we describe the changes to the genome that have been associated with defects in individuals, we can, and we have information to genetic and genomic information about parents and even embryos, there is a tendency to predict what will happen based on sequence alone. So we don't really know. So we know that in people in which there is a phenotype, there is a mutation. We have absolutely no way of predicting whether and in which cases the presence of a genetic modification will cause something in a human. So these are very old questions there, but they're desperately in need of new tools. And I will tell you about the work of our lab, in which we took one highly conserved signal transduction pathway that is initiated at the cell surface, propagates through a network of protein interactions and enzymatic reactions, and influences cellular functions such as proliferation, gene expression, cell shape changes, and so on and so forth. We took one node of this network. We took a family of mutations that have been identified in a class of specific developmental abnormalities that I will just describe, and we tried to model them in an organism that gives unique opportunities for combining old questions with quantitative measurements. So first of all, this class of developmental abnormalities is called rasopathies. Opathies is a class of diseases, and RAS is a name of a highly conserved signal transduction pathway that starts at the cell surface, typically with the activation of receptor tyrosine kinases, leads to membrane recruitment of certain proteins, eventually plugs into the three-stage enzymatic cascade where there are three kinases, RAF, MEK, and ERK, where RAF phosphorylates MEK, activates it, MEK phosphorylates ERK and activates it, and ERK has a large number of substrates in a cell, and in this way connects an event at the cell surface to changes in cell behaviors. Mutations in this pathway that arise in somatic tissues have long been known to be associated with cancers. So for example, if a person goes out in the sun and is exposed to UV light, a mutation, a DNA lesion can cause a mutation in the skin, and this is how melanomas start. Smoke that induces in the lung, number of mutant pathways have been described. It turns out that exactly the same proteins can be mutated. So, and the change to the organism, to the function of the adult organism, is localized to the place where mutation arises until the onset of metastatic spread. So it turns out that the same components can also be mutated in the male germline, specifically during spermatogenesis, giving competitive advantage to the future sperm cells. So if such a sperm cell is formed, and if it fertilizes the egg, and if this fertilized egg gives rise to the embryo, and this embryo develops, then mutation is present in every single cell of the organism. And because this signaling pathway, the RAS pathway, is involved in essentially every single aspect of embryonic development, there can be perturbations. So individuals with such mutations display developmental abnormalities that come under the umbrella name of rasopathies. So this is the term that was coined by Catherine Rowan, a US doctor, approximately 10 years ago. So individuals with these kinds of mutations display a syndrome. So they have facial malformations. Specifically, the faces are asymmetric. They have very specific congenital heart defects. They're typically shorter. They grow slower. They display delayed onset of puberty. There are significant deficits in intellectual abilities. So once again, this family of diseases is called rasopathies, and they come from the germline mutations in the RAS signaling pathway. So there are multiple components of this pathway, and mutation can arise in any single one of them. And in individuals that have been characterized, a single mutation can be traced to one or the other component of the signaling pathway. So about 10 years ago, two former members of my lab, Grant and Jindal and Yogesh Goyal, started thinking how to model such diseases in model organisms, such as flies and zebrafish. We thought long and hard which component of the pathway to select for our experiments, and we chose a component that is called MEK. MEK is an intermediate kinase in the RAF-MEK-ERK cascade. We chose MEK because it appeared to us, and still does, the simplest component of the signaling pathway. It is an enzyme that sits inactive inside a cell and awaits a signal from a cell surface receptor that is bound by an extracellular ligand. When this happens, there is a single kinase that activates MEK. This kinase is called RAF. When MEK is activated, MEK has a single substrate, which is called ERK. So there are two reactions that can be affected, and MEK can also be dephosphorylated. So that's one or two more reactions. So we took a component of the signal transduction pathway that participates in the smallest number of interactions, And then we took a whole bunch of mutations that have been identified in either human cancers and developmental abnormalities, and we asked what is the effect of different mutations on different steps of activation of MEK, and we could answer these questions using a combination of experiments in the test tube with bacterially expressed and purified components of the RAF MEK ERK cascade, or in experiments in embryos where either normal or mutant variants of MEK were overexpressed. So summarizing years of work, I can tell you that everything that we found can be summarized in 3 bullet points. So first of all, normal MEK is inactive until ligand comes to the cell. MEK that harbors these mutations is faster in responding to ligand. Furthermore, such MEK displays some level of ligand-independent activity. Third, this MEK requires a certain adapter to come together with RAF in order to be phosphorylated. Some of the mutants of MEK can be independent of adapters. So already in this, and different mutations influence these processes, ligand-independent activity, response to ligand, independence of adapters to different extents. So already see that different mutations can disrupt this pathway to different extents. There are lots of interesting questions in which we try to connect the 3-dimensional structural information about the molecules that are involved in the relevant processes to the changes in the rate constants. But today I want to focus on what are the consequences of. So this is the main question. So we know that defects. So today, a pediatrician, when they see a child with a combination of heart defects, certain facial features, delays in growth, they can suspect rasopathy. This suspicion can be confirmed by sequencing of the genome and associated with the mutation in a protein like MEK or RAF or RAS. So defects imply the presence of a mutation. So it is a necessary condition for a mutation. So one can, almost like in a math class, you can ask, is the presence of mutation sufficient to cause a phenotype? So the ideal experiment that one does in order to answer this question is that you would like to follow development in a large number of individuals that have identical genomes that are placed in identical environmental conditions, but have a single change in a genome. Specifically, we would like every protein to be normal, but we would like MEK to be expressed at normal levels, but we would like this protein, this kinase, to have a single amino acid modification. And we would like to do this experiment for multiple mutations of MEK. So such an experiment is obviously impossible in humans. It is very hard in mice, although it's becoming more and more doable. And it is very much possible in flies, as I will describe today. So Rob Marmion, a postdoc in our lab, after a number of years of trials and errors, established lines of flies in which the genome has a single modification. So there are 2 nucleotide changes. One of them is necessary to make the genome editing process to work, and the other one is the nucleotide modification in the coding part of MEK, and it can encode MEK where pretty much any amino acid that is normal altered to amino acid that has been associated with diseases. So, and we can establish these animals as either homozygous or heterozygous, which is relevant for studies of disease. So, first of all, in a human, we know that we have developmental delay, we have shorter stature, there is craniofacial morphology, heart defects, so on and so forth. What does it mean to model a defect in flies? So defects in craniofacial development, we know, and everybody knows, have to do with neural crest, the formation of neural crest and migration of neural crest cells. Well, insects do not have, invertebrates do not have neural crest. So how can we model this? Fly's heart is also very different from a human heart. The endocrine signaling in flies is similar to endocrine signaling in humans, but not identical. So when we say that we can model something in flies, what we mean by this is that we can look at the developmental processes in the fly embryo and in the fly larva or in the adult that are known to depend on the RAS signaling pathway, and we can examine the presence of defects. So today I can tell you that we succeeded in confirming that organisms that are homozygous or heterozygous for these mutations demonstrate developmental delay. The presence of these mutations leads to partial lethality. I will explain this. Their wings have extra veins, so humans do not have veins, but this is an example of a morphological defect. The eyes that depend on RAS signaling pathway, they are disrupted, and that's another RAS signaling pathway, and they also show several other phenotypes. So we have animals that present a complex of imperfections, very much like we have humans that present a complex of imperfections, presumably because of a modification in a specific component of a signaling pathways. But in flies, we know that this is the only change that is happening. So this is the first result that I want to tell you about. So it's from a paper that was published by Rob Marmion and other people in our lab in the middle of last year. So if you have a fertilized egg of a normal fly, of an embryo of a wild-type genotype, approximately 90% of these embryos after a day will give rise to larvae that will run. So when the embryo runs out of food, forms a larva that hatches from the egg and goes out to find new sources of food. So 10% of eggs that do not hatch correspond to imperfections or inefficiencies in the fertilization of the egg. So in other words, embryonic development it has a very high yield. Approximately 100% of them give rise to the larva. When we have embryos that are equivalent to the wild-type embryos, with the exception that they encode MEC that has a single amino acid change, and this is just one example, 50% of only 50% of embryos hatch. Those that do can hatch with a significant delay. So this means that the presence of a mutation can disrupt development in two different ways. It can either give rise to a development that gives rise to a larva, or it can completely kill a developmental trajectory. So this leads to the obvious question: what is it that is different between embryos that give rise to larvae and those that do not? So if you tell about such partially penetrant phenotype to a human geneticist or to geneticists in general, the first reaction is that most likely it's the presence of genetic modifiers. So we cannot rigorously exclude this possibility in our work, but we try to work with populations of inbred flies, but it is still possible. The second possibility is that there can be environmental factors. So embryos develop from eggs, and eggs can be slightly different in the amount of nutrients that were deposited by the mother into these eggs, nutrients or patterning molecules, such as localized RNAs. So maybe it is slight differences in the initial composition of the egg or slight differences in the place where the egg was developing on the surface of a dish, okay? Or maybe it is pure chance. So in the next couple of slides, I will try to show you that while we cannot completely exclude the effects of genetic modifiers, environmental factors, we can assign the partial penetrant phenotypes in our experiments to purely stochastic effects. Why is this a relevant question? The question is relevant because doctors that work with see individuals with this class of developmental abnormalities know that people with the same mutation can present functional and morphological defects at different levels of severity. So when there are completely different people, you can say, well, there are genetic modifiers, they have different parents, and maybe it's the modifier genes somehow mask or amplify the effect of a change of a specific signaling protein. So, however, it turns out that even monozygotic twins that have identical genomes can show differences in their developmental trajectories. So what you see on the left are X-rays showing where you see skeletons or skeletal structures of two sisters that are monozygotic twins. One of them has normal development, and the other one has scoliosis. And this is just one of the phenotypes that is abnormal in these individuals. So in these sisters, there is a mutation in a gene that is called NF1, which also belongs to the RAS signaling pathway. So differences in organismal development can be seen even in monozygotic twins. So when you tell human geneticists about such a phenotype in monozygotic twins, they will tell you, well, maybe there is some mosaicism in development of these twins. So that explains this. The embryos could have been definitely are implanted in different parts of the uterus. So this high variability in development that is caused by the presence of these mutations is known, but it's very difficult to get to the causal origins. So our approach in trying to tease apart systematic effects, which are modifier genes and environmental changes, and stochastic effect, had to do with looking at with using the fact that flies and us and many other animals are bilateral. In our structures that are identical on the left and the right hand side of the organism, such as paired ribs, paired muscles, and so on. And we work with dependent on signaling through the right. In comparing the formation dependent structure, both sides of them, we can be sure that the organism has the same, that we're doing comparison, keeping the genotype constant, and that the environment is constant, such that the embryo was developing in one fixed environment. Okay. So the phenotype that I will describe is the phenotype in the morphological structure that is present in a fruit fly larva. So the video that is just playing shows you a network of tubes that bring oxygen to tissues in a developing larva. Oxygen enters through spiracles that are present in the anterior and posterior of the larva. It flows through the stereotypic network of tubules and eventually comes to the distribution points, which are shown as these purple cells. So these purple cells can be thought of as shower heads that let water in your bathroom. So water is flowing through pipes that are not leaky, but only when you need water, you can open the faucet and let the oxygen and let the water flow. In this case, the entire surface of the larva is tiled with these cells that have very special architecture. So you are seeing one pair of such cells. So the cells are shown in blue. There is one cell on the left, one cell on the right. In both cells, you're supposed to see a white nucleus. I'm pointing at the nucleus in the cell on the left and the nucleus in the cell on the right. And this branched structure is just a single cell. So these cells have absolutely remarkable architecture. So these cells are unique in that they have oxygen in them. So normal cell, if you cut it, you would see plasma membrane, cytoplasm, and plasma membrane. So these cells, if you cut any of these, green structures transversely, you would first go through a plasma membrane, then through cytoplasm, then through a plasma membrane, then through oxygen, and then another plasma membrane, cytoplasm, plasma membrane. So they have the topology of a glove, and inside this glove is air. And I don't know how well it comes out on screen, but if you see the dark streak at the bottom here and the dark streaks here, you're actually seeing gas-filled lumen. So this is a schematic representation of the structures on two sides of the embryo. So one cell on the left, one cell on the right, and there are plenty of such cells on both sides of the organism. So each of these cells is selected in a RAS-dependent manner from a primordium that has 6 to 8 cells. So there is a signaling event that singles out one of these cells to assume this highly branched and gas-filled, highly branched morphology with a gas-filled lumen. When there is increased signaling, sometimes more than one cell is selected from the same primordium. So importantly, this is not a defect in cell proliferation. No new cells are generated, but from exactly the same primordium, more than one cell is selected to acquire this morphology. And we can easily detect the presence of these extra cells because they have such unique morphology, and they can be marked using fluorescent proteins. So a normal cell, in a normal larva, in a normal segment, there will be one cell on the left, one cell on the right. So here the left cell is highlighted in green and the right cell is in this magenta color. However, when there is extra signaling, we have extra cells. So we have one side of the organism that has extra cell and the other side of the organism that has extra cell. So this is very much like polydactyly, but the extra structure is not an extra finger with multiple cells, multiple tissues, nerves, and very complex architecture, but we have just a single cell that is formed in an extra. Okay. So what is important for our, so when this is done, so the organism is at the same genotype. The genotypes are the same on the left side of the larva, on the right side of the larva. There are multiple segments that give us an opportunity to rewatch this game replayed again and again and again. It is important that the phenotypes are not always symmetric. So in this case, there is only one cell on the right, but there are two cells on the left, one normal and one ectopic, one abnormally formed. And here is just a range observed in individuals that have the same genome of the organ. So the range from one cell on the left, one cell on the right, extra cell on one side of the organism, two cells on the left, one right, extras, and sometimes we even see 3 extra cells. Okay, so this is like having polydactyly that is not bilateral, where there would be an extra digit on the left, 5 finger. Doing statistical tests of the occurrence of these defects in the left side of the organism and on the right side of the organism, we convinced ourselves that a random model is sufficient to explain what we observed statistically. It was a very simple statistical test. And Lena Barrett and Alison Simpkins, the 2 students who did these experiments, have collected a large number of observations from a large number of larvae. And they have done this in multiple genotypes. And we think that this is an example of a defect that comes from, that cannot be assigned to a modifier gene and that cannot be assigned to differences in environment. So the main thing that we learned from this is that the presence of a mutation is not a sufficient condition for causing a phenotype. The phenotypes are variable, they can have stochastic origins. And we are now following up this work by imaging the emergence of these defects live. So we managed to narrow down the developmental window where these phenotypes, where these extra cells arise, to 1 or 2 hours. And we want to detect the number of ways in which these structural abnormalities can arise. We're quantifying the changes of signaling that can give rise to these abnormalities. Based on our preliminary data, we think that these perturbations are really mild. So specifically, the level of pathway activation, appropriately defined, is affected only to 10–15%. We want to understand what is it that makes this specific stage of development so sensitive. Okay, so at this point, Joel, can you tell me, I don't have a clock here, how am I doing on time?
So it's 35 past, so another, I'd say 15 minutes would be
okay, very good. All right. So in this case, in telling you about the Drosophila modeling of human developmental abnormalities that come from permanent modifications in the genome, we see what can happen when perturbation to the signaling pathway is present all the time. So now I will tell you about the perturbation of exactly the same signaling pathway. So the molecules will be exactly the same, MEK and ERK, but we'll be perturbing them in an acute manner, trying to understand how the organism responds to perturbations that are not static, but are acute. Okay, so MEK, the kinase that we were studying in the work that I have just described, is very, very specific. It has a single substrate, and the substrate is a kinase called ERK. ERK, on the other hand, has approximately 1,000 substrates. So we don't really know how many substrates are present in any given stage of development, but if you compile all the literature that has been collected about ERK in different organisms, from flies and snails to frogs and humans, you will find out a very large number of substrates. So the question that one can ask for such broad specificity kinases is that how many of the substrates are important for any given stage of development? How many of the substrates are actually phosphorylated? How many of these phosphorylations are functionally significant? Again, this is an old question, but only now we start having tools that can be used to probe this question systematically in the living organism. Okay, so the stage of development that I will tell you about is Drosophila embryo, when it is still very young. So the movie that you are watching now is the fly embryo that is 2 hours, that gets to the age of 2 hours. And this embryo goes through a sequence of sequential and highly synchronous cleavage divisions, where the number of cells goes from 1 to 2 to 4 to 8, all the way to 2 to the 13th. So eventually, in a process that is very interesting, most of the nuclei end up under the common cytoplasm in the embryo, and they divide 4 times in a relatively synchronous way, presenting a very interesting system where you can study signal transduction simultaneous of cells that have exactly the same genotype and have exactly the same developmental age. Okay. So after these divisions have happened and you have a monolayer of cells, monolayer of cells is patterned by signals that ultimately trace to the systematic non-uniformities of the egg that were established during oogenesis. Specifically, there are very well-defined non-uniformities in the composition of the extracellular matrix at the anterior and posterior poles of the embryo that give rise to the activation of the ERK signaling pathway, the same signaling pathway that we have been studying when we looked at MEC. So ERK, which is activated in response to localized stimulation of receptor tyrosine kinase pathway, is activated and is known to phosphorylate and negatively regulate a transcription factor that acts as a repressor of genes that are needed to specify the most anterior and the most posterior parts of the embryo. So here only one of these genes is shown here in magenta. So ERK controls patterning of the anterior and posterior poles of the embryo by phosphorylation of a protein that is called capicoa. And based on genetics, capicoa is either a single transcription, single substrate of ERK that is necessary for this modification, or one of 2 or 3 or 4. Okay, so this is an example of a case where there is localized activation of a signaling pathway, and its role in patterning can be assigned to a very small number of substrates. But remember, I told you that ERK can have a very large number of substrates. So what is actually happening here? It is either somehow it happens that no other substrates are affected by ERK, or they are affected and are not important for biological function, or we are missing some functional understanding of ERK signaling pathway. So maybe it's doing something else that cannot be detected or was missed when people were using assays and fixed embryos. Okay, so the way we decided to probe this is that we decided to activate this pathway very acutely using optogenetics, using a technique that was established by Heath Johnson and Yogesh Gayal. Heath was a postdoc in the lab of Jared Tocher, and Yogesh Gayal was a student in my lab. So they used the technique that was initially developed by Jared Tocher during his postdoctoral work that allows you to use blue light to very rapidly recruit a key component of the signaling pathway to the plasma membrane and stimulate the pathway in a way that's independent of growth factors. So under the normal course of development, this pathway is not active at all in the middle of the embryo. So the level of activation is 0, is activated in the anterior pole and is activated in the posterior pole. So in response to, so what is important is that this activation does not leak. If you keep your flies in the dark, the embryo is not stimulated. But if you expose the embryo to a 10-minute pulse of blue light, you activate the pathway throughout the embryo in a more or less uniform way. We can talk about non-uniformities if someone has questions. But it's very important that even the highest level of activation is comparable to what the embryo is normally exposed to. So because most of the embryo under normal conditions is not activated, this uniform activation gives rise to a very large change in the average level of pathway activation in the embryo. Because there is no limit to the number of embryos that can be collected in response to the stimulation, one can compare the global phosphorylation changes in a normal embryo and in an embryo that received a 10-minute pulse of blue light. So then, in collaboration with the lab of Martin Wu, my colleague and neighbor in the Genomics Institute, Liu Yang, a postdoc in the lab, and Audrey Zhou, a graduate student, have collected and compared the phosphoproteins of embryos that are normal and that receive a 10-minute pulse of blue light. So this is an embryo that developed absolutely normally for 2 hours of its development and then received the 10-minute pulse of its light. And then the embryos that were stimulated and received acute perturbation and those that were left in the dark were lysed, and their phosphoproteins were compared. And everything was done here very carefully after a number of years in which Leo and Audrey were developing protocols. Each condition was analyzed in 8 replicates. So we found a large number of changes. So Capicoa, the substrate that we know to be phosphorylated, responds. ERK responds because we activated it in response to light. But we also found a large number of proteins that were phosphorylated. Some of them were dephosphorylated, indicating that ERK induces a very large phosphoproteomic change in the cell, even when it is activated to the levels that correspond to normal levels of pathway activation in the embryo. So we are still making our way through analyzing the data, but the most important finding is that a large number of substrates seem to be associated with control of the cell cycle that have to do with assembly of mitotic spindle, centrosome duplication, breakdown of the nuclear envelope. So based on the functional annotations of the substrate that came from phosphoproteomic analysis of embryos that received acute ERK perturbation, we asked whether this is something that this pathway is actually doing normally. And I will give you 2 pieces of evidence that prove that this is actually the case. So the first piece of evidence comes from disrupting the normal course of ERK activation in the embryo and watching cell cycles. So normally, all components of the pathway are present uniformly throughout the embryo. The pathway is activated only locally at the poles. So I will play 2 movies simultaneously. First of all, let's watch them, and then I will explain to you what you're supposed to see. All right, so I hope the movie is played. And what you're supposed to notice is that divisions in the top embryo are very synchronous and fast, and the divisions in the bottom embryo are less synchronous. So in the top embryo, there is a fast wave that sweeps through the embryo. In the bottom embryo, the cell cycles seem to be much more out of sync. So based on this, and based on the previous information about the role of ERK in controlling cell cycles in frogs and in mammalian cells, we hypothesized that ERK accelerates cells, that feeds into cell cycle progression at the poles of the embryo and communicates this effect to the rest of the syncytial blastoderm. So in this case, these nuclei sit in the shared cytoplasm. So there is a possibility of diffusion and other sorts of communication between different parts of the embryo by molecules that are involved in controlling different parts of cell cycle. So if this is the case, and if ERK activation at the poles of the embryo acts as a conductor that coordinates the synchrony of embryonic cell cycles, then one should be able to supply a pulse of ERK activation in the center of the embryo and initiate a wave. So this is a movie in which nuclei that enter mitosis, or cells that enter mitosis, are colored in yellow. Cells in the interface are colored in blue. And you see that there is very rapid initiation and propagation of mitotic waves through the embryo, and that mitotic waves typically initiate at one of the two poles. And in this experiment, Liu shines light in the middle of the embryo. So the early divisions are normal, they're synchronous. And then as cell cycles slow down, we will see robustly in these embryos something that is never ever observed under normal conditions. You will now see a wave that starts from the center and propagates to the poles. So by disrupting normal progression of the pathway, we lose synchrony. By providing ectopic pathway activation in the center of the embryo, we can start light-activated propagation of the wave. So based on this, we believe that this pathway has a new role in the embryo. In addition to controlling transcription, it is also controlling synchronous formation of a cellular layer that needs to respond to patterning signals that ultimately control transcription. So to summarize, I told you today about our work that is motivated by the need to understand developmental defects. Ultimately, we think that quantitative understanding of developmental effects will come from quantitative studies of developmental systems to perturbations. And different types of perturbations are different for answering different types of questions. Permanent perturbations can tell you how defects in one stage of development can influence the others. And optogenetics can be used to understand what are all the functions that the activation of the signaling pathway plays at a given stage of development. And that's it for today. I'm happy to answer questions if there are any.
Thank you for the talk. Do we have any questions? Either post them in the chat, or you can put your video on and unmute and ask them directly.
Okay. Hi, I'm Mathieu Emery. I'm essentially a research engineer, but I have a strange question about the fraction of larvae that do not hatch, the eggs that do not give a larva. Because it seems that even for the non-mutated one, you still have far less, of course, but like 5 or 6% of eggs that do not hatch. Do you think it's the same mechanism for both, just overexpressed for the mutant?
Okay, no, it's a completely different. It's a completely different effect. So what I tried to say, but apparently did not come out clearly, is that the ones that do not hatch without mutation, these are eggs that were not fertilized. So here, embryonic development did not even start. And when there is a mutation that is active, development can get off rails at multiple points. So we can normalize the develop. So we can take the 90% of normal embryos as normal, and it's a completely different mechanism. Yeah. Okay. Shall I?
I've got a question. So for the light activated ERK wave starting from the center, like how did development proceed after you did this perturbation?
Okay, so the embryo is killed because in addition to controlling cell cycle, or the ERK pathway is also controlling patterning. So you are disrupting gene expression in the center of the embryo. And because of this, there are severe segmentation defects. You disrupt the formation of the ventral furrow. And we think that the reason that the cell cycle effect was missed before is that, first of all, the embryos where the ERK pathway, because the original genetic screens were based on the phenotypes of cuticles, and you look at the final outcome. And what the embryo wants to do, it wants to establish a blastoderm, an unpatterned blastoderm, as quickly as possible and get it patterned. And when you're examining phenotypes that come only later on, you can miss defects that can be corrected for.
So could there be any like if there was like a smaller perturbation with a different pattern, do you think it would still be possible for development to proceed?
Yes, absolutely. So in this case, the light was shining on the embryo for an hour.
Mm-hmm.
So we haven't narrowed it down to a smaller perturbation. So in this experiment, we just showed a sufficiency. So we know for a fact that localized disruption of the activation pattern of this pathway gives rise to a defect in gene expression. This was known long ago. What we show here is that in addition, but it was defect in the patterning that was of a tissue that was already established. So here we're showing that even before defects in patterning, we have a defect in the synchrony of the formation of the cluster.
Mm-hmm. Cool. All right, so we have a few questions from the chat. Guilherme Ventura asked, for how long are the waves sustained when you optogenetically activate ERK in the center of the embryo?
Okay, so these waves have the way that the embryo is developed, it is fertilized, and in 2 hours it goes through 13 cleavages. We can perturb the system when we can image it. So we can perturb it even earlier, but we want to understand the effects of perturbations on the stages that we can image. This is done during the last hour of development, and these waves are transient. So at some point, so these are waves of divisions, and at some point the embryo will stop dividing. So we can disrupt the synchrony of 13 divisions that happen, but we do not affect the number of divisions. In some cases, that can be an extra division, but it's a separate story.
Okay.
Okay, I see that there is a question of the fact from the chat, how does ERK control the cell cycle? So in this case, we have preliminary data that are pretty convincing that this depends on the phosphorylation of protein that is called CDC25, which is a phosphatase that removes inhibitory tyrosines from CDK kinase, which is a master regulator of mitosis.
And then I guess the other questions in the chat are, can this optogenetic tool be used to study other signaling pathways in the Drosophila embryo? And what would be the prerequisites for studying a signaling pathway with this tool?
Okay. So this is a tool that is based on the design that Jared Tocher came up with more than 10 years ago that has to do with the recruitment of a constitutively active component to the plasma membrane. So by now, several processes in the embryo and other signaling pathways have been placed under optogenetic control. You can, for example, take transcription factors and bring them in and out of the nucleus, as was done in the work of Eileen Furlong with twist and snail. So this was done in the MBL. Other signaling pathways, I think the BMP signaling pathway that controls the dorsal patterning of the embryo can also be placed under optogenetic control. This has been done in fish. I'm not sure if it was done. It has been done in the fly. It's a very rapidly developing field. So it's not guaranteed that whatever design you take from a cultured cell or in the fly organism will work from another organism will work in the fly, but a lot of things can be done. So you need to have a constitutively active component that that has kind of latent activity that you can activate or by light.
Mm-hmm.
Yeah.
Cool. So Hao-Chi Chen has the question, how do you unify the two functions of ERK, namely the controlling of patterning, which requires differential ERK activity level, and the synchronization of the cell division, which requires uniform ERK activity?
Yes.
Into
Okay.
one
So
model?
in this case, so I will answer this question and then I need to run because I need to go to a talk of a faculty candidate. So in this case, this can actually be done using a separation of time scales because the cleavage, so so when you go through divisions, 2, 3, 4, and so on and so forth, these divisions are so fast that there is not enough time for transcription to take place. So we think that there is an earlier part that has to do with control of the formation of a field of nuclei. During this time, there is no transcription. And then as cell cycles get slower, there's actually enough time to transcribe zygotic gene products. So this would require a model in which there are coupled cells or coupled oscillators that change in number that depend on their synchrony on the localized conductor. Okay, guys, I need to go to another building and it's quite cold outside. I don't want to miss the talk of the faculty candidates. Thank you for having me.
Thank you so much for joining. Have a good day.
Happy New Year to everyone.
All right, thank you. And thank you to everyone else for coming to the talk today. All right, bye.